I'm an epidemiologist, so I'm going to talk about the sort of opportunities for epidemiology from genomics. So that means that the few of you who aren't doing your email can go back to checking your email for 10 minutes. A major contribution of genetics to epidemiology is that it can help strengthen causal inference with respect to modifiable exposures. So the interest for epidemiology is things you can change with environmental changes. So the trick of this is that you have to make the assumption of G equals E, i.e. gene-environment equivalence, which is a commonplace in developmental biology. But so you have to have an idea about whether that is reasonable. So, for example, if you're looking at cholesterol levels, LDL cholesterol as the modifiable exposure, is it reasonable to assume that the effects of cholesterol on coronary heart disease will be the same if they're influenced by the environment, diet, cholesterol-lowering medication, etc., or by your genetic variants? Now, in this case, it is reasonable to assume that's the case. NPC1L1 variant, for example, relates to cholesterol transport across the gut, and the transport of cholesterol from diet and from bile across the gut. So it's reasonable to think that is mimicking the same thing. And of course, there's a lot of genetic variants which straightforwardly mimic statins, PCSK9 inhibitors, NPC1L1 drugs, etc. But so as long as you can actually defend that notion of gene-environment equivalence, that does allow you to make inference about the causal effects of LDL cholesterol on the outcome. The model is an instrumental variable model, a simple model where you can actually demonstrate that your genetic variant associates with the intermediate phenotype. That's the one thing you can demonstrate. You can't actually have a test for the fact that confounders don't relate to your genetic variant. In genetics, of course, and this is a distribution of genetic variants, the main confounder will be population stratification, which you will deal with, and you can look at whether the variant relates to confounders, but you cannot test that they are unassociated. The third is the exclusion restriction, that the genetic variant doesn't have an effect on your outcome except through the intermediate phenotype you're interested in. This is, of course, assuming no horizontal pleiotropy, i.e. pleiotropy, which is not through your intermediate phenotype. You can't test that. And one thing you absolutely can't do is you cannot test that by adjusting for the intermediate phenotype, because if you do that, that then means that all the power of the design breaks down because the confounders become related to your genotype. That is referred to in the causal inference world as adjusting for a collider and leads to confounding where there is no confounding. So the one thing to remember is that you cannot test the second two assumptions. If you think you can, then either you deserve a Nobel Prize or you're wrong. And so far, every case, I've reviewed many papers saying they're testing this. All of them so far have been wrong, but I look forward to your Nobel Prize-winning paper. Now, horizontal pleiotropy is, of course, the major problem. What can we do about that? Well, there's a whole host of sensitivity analyses. And the important point about these are that they all have assumptions which may very well be wrong. But for many of them, the assumptions are absolutely orthogonal, i.e., if one of those assumptions, if assumption breaks down for one method, there's no reason why it should break down for the other method. This includes over-identification tests when you have many genetic variants, and you can demonstrate whether it's plausible that they'll all be confounded or all be influenced by horizontal pleiotropy in the same way. Negative control, no relevance points. For example, in East Asian populations, looking at a genetic variant, ALDH2, that relates to alcohol consumption amongst women, and when women are not drinking, but the horizontal pleiotropy should be revealed. There is then, between siblings, Mendelian randomization introduced years ago, when the Mendelian randomization is carried out between siblings, and that will not be biased by assortative mating, by non-transmitted allele effects or dynastic effects, as they've been called in economics. M.R. Egger, which makes the assumption that the pleiotropic effects do not correlate perfectly with the effect on the exposure of interest. Anyway, the point is that all of these make different assumptions. Not all of them. Many of them make different assumptions. Some of them group together. So, for example, Cook's distance, Mr. Presso, glide, and the constrained IV methods are essentially outlier detectors and exclusion of outliers. but many of these methods have completely orthogonal biases. The interaction approach is one which also allows estimation. When we introduced this in the MR context 10 years or so ago, we made the assumption that in the women who were not drinking in these populations, that was a no relevance point. The instrument was not associated with the exposure in a group who don't have the exposure, which in this case is alcohol. But you can relax that, and this is a really powerful, as yet little used approach. When you have different groups which have different levels of exposure, then the regression is your causal estimate, and the offset is your bias term. So for individual variants, you can actually demonstrate whether pleiotropy exists for individual variants. Many of you will notice this graph looks rather similar to MR-EGR. It looks similar to LD score regression, all of which use the offset term as a bias term, as did actually the original EGR regression 25 years ago for meta-analysis. But the use of offset terms as bias indicators and slopes as effect estimators is, I think, powerful. And now with massive sample sizes with individual level data, there's real opportunities for using this approach. So you can triangulate across genetic approaches. What is most powerful is when you can relate a non-genetic causal and analytical strategy to a genetic analytical strategy. So here is one example of that. This is biobank data on the effect of one additional year of school education. In 1971 in the UK, people were forced to stay in school for one extra year. That led to an average 6-month increase in educational time for people in schools. I was the cohort after that, 1 year after that. To me, it seemed that the whole purpose of that additional year for these kids who didn't want to stay in school was so they could stay there and beat me up. The idea that this could have any positive effect on anyone, to me, seemed remarkable. But of course, you can very easily see what you can do as a regression displacement, taking that the cohort who went through the period in 1971 before compared to after, and compare the regression lines. The remarkable thing is carrying out Mendelian randomization, which is in blue, and using this as the minimum school leaving age instrument, and then versus the observational data, you get a coherent pattern. The first is the one you absolutely expect, which is you get larger effect sizes with instrumental variables because they are measurement error corrected. That's why you get 2.5 times the effect estimate in Mendelian randomization studies of LDL cholesterol, because it reflects to LDL cholesterol over 60 years of life, not the 5 years in a statin trial. So they're measurement error corrected, and they show large beneficial effects of education on many health outcomes and on social outcomes. So when you triangulate there, it is very obvious that those biases are orthogonal. There's no reason why being born a day apart in 1971 is anyway related to your genetic variance. That's very, very obvious. Multivariable MR, I think, is a powerful tool which has been recently enhanced. And this allows you to potentially sort out highly correlated phenotypes. So, you might have a situation where you've got a confounder situation where you're interested in this phenotype, but another phenotype, say body mass index, is influencing this phenotype and is confounding it. So multivariable MR, when you apply a set of genetic instruments that relate to those two phenotypes, can sort that out. The real magic of multivariable MR is it is not susceptible to collider bias because you use the predicted genetic effect, not the actual phenotype. If you adjust for the phenotype, you introduce collider bias. If you actually use the genetic predicted effects, you do not. There's obviously several models which you could apply to such data. Another one is horizontal pleiotropy, which I've already discussed, and a final one is the mediation, where the variable is actually influencing it through another variable, but hasn't influenced itself, as does the other variable. Now, mediation and confounding, you'll notice, are mathematically the same. So one of the interesting things about, if you just apply an analysis, a naive observational analysis to data, you can't separate mediation from confounding. You have to have some prior causal knowledge which allows you to make that separation. So let's take one last example, which is educational attainment, cognitive ability, and smoking behavior. You know, two potential DAGs, there are many, or directed acyclic graphs, the educational attainment influencing cognitive ability, or cognitive ability influences educational attainment, which is the one which seems most reasonable. I apologise for the next slide, I should have made a decent slide, but the results, again, within UK Biobank on this analysis are that both, this is with smoking as your outcome, that both educational attainment and cognitive ability influence your smoking. That's your ordinary regression. Luckily, the regression is larger in the IV analysis, as it should be, because, of course, you're correcting for the measurement error, which is considerable in these variables. But then the multivariable MR suggests that it's educational attainment which influences smoking. Taking that into account, cognitive function has little effect, which makes some sense. But I'm deliberately using a sort of social example because it's in a way the most problematic. You then do between siblings analysis to take into account non-transmitted alleles, assortative mating, etc. You do all the other sensitivity analyses on this. You'll be pleased to know if you look at other factors with voluntary physical activity, engaging in physical activity, education is related to a considerably higher level of it. Conditional on education, one more slide. Conditional education, cognitive function means you do less, which makes complete sense to me, because if you haven't been told it's good for you, you get some other sod to do the hard work. Anyway, so you can sort out these correlated factors. You'll recognize the multivariable model as being the omnigenic model. The omnigenic model, as it's finally been formulated in the Zhu et al. Paper, defines it explicitly as that conditional on the genotype and expression levels of all core genes, the genotypes and expression levels of peripheral genes no longer matter. You can test this within a Mendelian randomization framework. If you try and just do the adjustments for the actual measured phenotypes, you will introduce collider biases. So the Mendelian randomization would allow you to test the omnigenic model. It is testable in that way. And with the last slide, I'm going to finish with something slightly different. This really is my last slide. I do think that we should also consider the major contribution of stochastic events and bounds to personalized prevention. And I'm going to go through one very recent paper that brought my attention back to this on cancers of bilateral organs. I first read about this as a medical student in a throwaway line from Richard Doll. I was a medical student in Oxford, about cancer of bilateral organs, which I think was in a 1971 paper of his. But it really does bring home the importance of stochastic levels, stochastic events. If you consider renal cell carcinoma, something which is a 15-fold difference between reasonable registries, cancer registries in different places, obviously huge environmental effect. And is increasing in Britain. Renal cell carcinoma, if you've got two kidneys, the germline genetic variation is, of course, null between the kidneys. They've got the same, except a few very early somatic mutations, they've got the same germline genome. Their exposure to your fruit and veg intake, your smoking, your alcohol, your physical activity and BMI, these are factors all of which seem to increase parenchymal tumours, are the same. Yet, if you develop a tumour on one side, your risk of developing your other kidney, developing a primary parenchymal renal cell carcinoma tumour is elevated, but not elevated by much. And now with longer survival, you can demonstrate how little it is elevated, as is the case for breast cancer, for example. So imagine, the perfect epidemiological study, which measures every single exposure you can known to man. They're not going to be different between the two tumours, or using germline genetics. So there's clearly just a huge stochastic element. And I think that insight from epidemiology is perhaps one that's worth considering. Thanks very much.
You that nobody's going to beat you up, and if anybody's trying to beat up George, I will have words with you. So that was wonderful, and really sort of an overview of what you're working on. So are there any questions for George? One or two? Yes.
Well,
Could you introduce yourself and your affiliation?
Richard Peto.
Who's not supposed to be here, so if he asks me a difficult question, I'll... Sorry.
No, I think Richard Dole's phrase for that cancer arises from nature, nurture,
From chance,
luck.
absolutely.
It's quite a good thing that, you know, given nature and nurture, you may have thousands of stem cells in your lungs, all of which got the same nutrition, exercise, and exposure to smoke. And it's just luck that one just happens to go to cancer and the other 1,000 that were similarly exposed don't. So you can illustrate it with the 2 lungs, but you could also illustrate it with the sort of hundreds or thousands of stem cells within one lung.
Absolutely. No, absolutely. No, the issue is that this effect is clearly going to exist for everything which isn't bilateral. It's just that you can see it. It's just an illustration. Yeah, no, I absolutely agree with that. And that's one reason why group level or population level influences that are driving the 15-fold difference between places will not be or need not be the same as the factors which are actually explaining why one individual develops cancer and the other doesn't.
One cell within one individual.
One cell, absolutely. It's the aggregate. What influences the aggregate might be different to what influences the lower level.